Hello everyone. So I'm happy to be here today to share big sessions on NK cells and the use of NK cells in therapy and particularly to treat hematological malignancies. So today we are very lucky to have one of the two best experts in the world working on NK cells and use NK cells for therapy. So I will start first by introducing Katy Rezvani, who is MD PhD and professor of medicine at the University of Texas MD Anderson Cancer Center. And she is also chief of the section for cellular therapy, director of translational research, and medical director of a good medical practice facility in MD Anderson. And she is also executive director of MD Anderson Adoptive cell therapy platform. And she is very well known for her research in her lab, focusing on NK cell biology and developing novel NK cells engineering strategy for cancer. And she wants to translate, and she has done already, translate his basic science research toward clinical work. And she completed her medical training at the Imperial College of London, and then she moved to the NIH for studying transplantation and immunology. And then she moved to MD Anderson. So currently she has published a lot of papers and very high quality paper, among 250 paper, and she received a lot of prizes and awards, and among them the American Society of Hematology Donnall Thomas Award, which is one of the most prestigious awards for cell therapy. So now with Katy, we have also Eric Vivier, who is a world-known scientist and for his seminal work on NK cell biology, and is a veterinarian as a education, and then he did his PhD at the University Paris-Saclay in France, and then developed a postdoctoral study at Harvard Medical School in Boston for 4 years, from 1999 to 93. And he make a lot of good study on NK, and we talk about that today. And he opened a very strong research laboratory at the CIML in Marseille on innate lymphoid cells. He's also professor of immunology at Marseille Medical School and assistance publique des hôpitaux de Marseille, where he's leading also a translational laboratory. And in addition, he served as director of the Marseille Immunopole, and is a chief scientific officer at Itnet Pharma, who is one of the co-founders of this biotechnology. And currently, and more recently, he serves as the president of the Paris-Saclay Cancer Cluster, and is a professor at École Polytechnique in Palaiseau. So now that I introduce these two prestigious scientists on NK cells, I would like, and today we will talk how to use these NK cells for treating cancer and particularly hematological malignancies. And we have two kinds of possibilities: using the cells themselves, or body to engage and activate these cells to kill cancer. So I will start first by basic definition, and I'm a great pleasure to ask Eric to tell us a little bit more on the biology and what are the peculiarities of NK cells. So Eric, the floor is yours.
Thank you very much, Olivier, for the kind words. It's really an honor to be invited tonight, this evening, I should say, and this morning for Katy. And it's great to have this talk with Katy. It's a great honor for me as well to talk with her. So thanks for the question, Olivier. Yes, maybe we can start by trying to define NK cells. And maybe if I can have the next slide. After this one and the one after again, I will start to try to recapitulate for you what are NK cells within the immune system. So first of all, NK cells are lymphocytes. They are very peculiar lymphocytes because they are lymphocytes of the innate immune system. So they are not T cells, they are not B cells. Actually, they were initially named as non-T, non-B cell, which is not a good start for a cell type. But over the last 50 years, because NK cells have been discovered 50 years ago, one has understood that there is a specific role for these cells, which is to detect stress cells. And I think this is extremely peculiar because they do not detect MHC peptide complexes. They do not detect all the antigens as B cells and T cells can do, but they are equipped with a variety of activating receptors that can recognize molecules which are induced by stress. And so when I'm talking about stress, I'm talking about stress induced by microbial infection, stress induced by cytokine stimulation, stress that can be induced by very stressing conditions such as hypoxia or metabolic stress, and obviously for our subject today, also stress cells which have undergone malignant transformation. And so the beauty of this recognition system resides in the fact that the molecules that are detected by NK cell activating receptors are most of the time not expressed at all or expressed at very low level in normal cell in steady state conditions. I think a prototypical example of this are the ligand of NKG2D. So NKG2D is an activating receptor expressed on NK cells. It's also expressed on subset of T cells, by the way. And the ligands for this NKG2D activating receptor are not expressed at steady state in most circumstances. But upon stress, and again, different kind of stress, including malignant transformation, then the cells can express a variety of NKG2D ligand. And it's easy to understand that through this mechanism, NK cells can detect stress cells in a very preferential manner or even specific manner. So that's the first point out of these 10 hallmarks of NK cell tumor immunity that I wanted to highlight. The second point that I would like to highlight in the context of cancer and hematologic malignancies is the fact that NK cells do not only recognize primary tumors, and here it's more on the solid tumor side, but they can also recognize and eliminate metastases. And I think this is a property of NK cells that needs to be more understood in order to harness this very interesting property. And finally, and to finish this slide, I would like also to emphasize that 20 years of NK cell infusion in patients, even in allogenic settings, I should say in particular in allogenic settings, have revealed that NK cells infusions are very safe. And the beautiful work of Katy, and I'm sure that she will present some of her results on that, on CAR NK cells have shown that NK cells can control hematologic malignancies. So this is just to give you a flavor of what NK cells are in general. But now if I can switch to the second slide.
Eric, may I
yes?
interrupt you here? Because you tell us that NK cells recognize stress cells.
Mm-hmm.
Can you explain to us what is the differences between T cells, NK cells, and invariant NK cells or ELC, for example? Because sometimes it's confusing for people who are not specialists.
Okay. Yeah, thanks for the question. Indeed. So there is, so to recapitulate, the world of lymphocytes, it can be divided into two categories: B cells and T cells on one side, and innate lymphoid cells. So innate lymphoid cells include NK cells, which are present in the blood, but also can infiltrate different tissues. Actually, one can find NK cells almost everywhere, not necessarily in high amount, but they can be there, and they can infiltrate the tissue in some pathological conditions, including tumors, but also inflammatory processes. But besides NK cells, within the ILC group, the innate lymphoid cell group, there are also ILC1s, which are very close to NK cells indeed. They can be cytolytic, as NK cells are, but they also produce a lot of gamma interferon, as NK cells do. And they have a lot of similar phenotypic features. But besides ILC1s, there are ILC2s, which are the innate counterpart of Th2 cells, and ILC3s, which are the innate counterpart of Th17, Th22. One of the big feature of these ILCs 1, 2, and 3 is that most of the time these cells are tissue resident. Actually, there are papers, including some of our work, that show that the progenitors of these ILCs resides in the tissue itself. We have shown in particular that in the liver ILC1s are originating from progenitors in the liver. They are not originating from bone marrow precursors as NK cells do. So to recapitulate, there are T cells and B cells that can recognize antigen in different forms. Obviously, antigen with MHC in complexes for T cells and other kind of antigens for B cells. But then you have ILCs, which include NK cells, present in the blood and elsewhere, and then ILC1, 2, and 3, which are mainly tissue resident. There is a fifth group of ILCs that I would like to mention, just for the information. It's called LTIs. These cells are particularly important during embryogenesis because they are participating, they are actually mandatory for the formation of most secondary lymphoid organs. So
and
that is, get your point, Olivier?
Yeah, very, very nice. You mean all these cells recognize some stressful cells?
Okay, so T cells and B cells, as we all know, can recognize everything, at least before selection. After selection, that's another story. ILC1, 2, and 3s are also equipped by this kind of activating receptors that I mentioned, some of them being shared by NK cells. In particular, ILC1s also express NKp46. Most subset of ILC1 express NKp46. NKp46, a molecule that we will talk later on again, is an activating receptor expressed on NK cells. It has been shown recently also to this idea of recognizing stress cells. So NKp46 has been shown to recognize cell surface reticulin. So we know that ectocalreticulin is most of the time expressed in the ER, but in stress condition can be expressed as a cell surface. NKp46, an activating receptor shared between ILC1 and NK cells, can recognize ectocalreticulin. So again, yes, you're right. These ILCs, NK cells including, have the propensity to recognize different forms of stress. To finish on that, they are really different from NKT cells. I would like to remind you that NKT cells are T cells, which express an invariant T cell receptor or semi-invariant T cell receptor. They are themselves close to mate cells, which are another category of, let's say, non-conventional T cells, but this is another story beyond the scope of today's presentation.
Okay, thank you very much.
So I wanted to to wrap up this first introduction on what NK cells are. I wanted to move forward 2 slides, if it's possible, Juliette. Okay, and stop at this slide. No, just the slide before this one. Thank you very much. I'm going to be extremely brief here, just to try to convey a very simple message. NK cells are not born all equal. NK cells are heterogeneous, and thanks to a collaboration with 20 different NK labs across the world, we have been able, through a meta-analysis of single-cell immunoseq data and site-seq data, to propose that NK cells are coming in 3 flavors, as represented here: NK1, NK2, and NK3s. Actually, NK1 and NK2s are not only present in humans, but also in the mouse. The work remains to be done to find out whether NK3s can be also found in the mouse. This is work in progress. But those subset of NK cells are quite different, and I would like to highlight this by the propensity to secrete cytokines. Because NK cells are not only killer cells, they are also cells that can secrete cytokines and chemokines that participate to the onset and the maintenance and the shaping of a T cell response. And this is here highlighted by the fact that the major subset of circulating human NK cells, the NK1, secretes CCL3 and CCL4, but you can identified here, you can appreciate that the subset of NK2s, a smaller subset in the blood, 10% of NK cells roughly, secrete XCL1, XCL2, which are very important cytokines, or chemokines, I should say, that participate to the activation and priming of CDC1, which are themselves so important in T-cell mediated tumor immunity. And finally, you can appreciate that NK3s have a very different cytokinic profile, as they are specifically or preferentially secrete CCL5 and IL-32. So I will just stop here. I hope I was not too long to try to describe both the commonalities between all NK cell subsets across mammalian species, but also the differences between the major subset of NK cells.
Okay, so and I have always a question, because there is some paper showing that NK cells, as you said, recognize some stressful cells because they do express some ligand from some NK receptors. And some people claim that although they cannot induce some kind of NK diversity due to TCR rearrangement because they have no TCR, but do you think that the panel of expression of different NK receptors may specify some kind of response against some type of tumors or some kind of virus or bacteria or so on? And we can get some kind of a memory of NK cells, which is some philosophical questions and
Well, I would love to have Katy's insight on these questions as well. But to begin the discussion, there are, I think, two questions in what you are mentioning. First of all, the point on the heterogeneity. Yes, based on the differential expression of several cell surface receptors. I have no time to go into the details, but yes, one can discriminate further subset beyond NK1, NK2, NK3s. But this can be, at the end of the day, an endless process because every single cell can be different to some extent. But again, please remember that the three major subsets, at least in humans, are NK1, NK2, NK3s. They have different expression at the cell surface of these receptors that convey indeed different function. I just focused on the former slide on the secretion of cytokines, but I could have focused on the differential expression of cell surface receptors. So you're right. So there is a differential repertoire of recognition according to the different subset of NK cells. Now, the second question in your question is about NK cell memory. So it has been a big debate, which started basically 15 years ago, starting from experiments using mouse cytomegalovirus in murine model, obviously, and showing that indeed there is some kind of clonal expansion of a specific subset of NK cells, which express an activating receptor, namely LY49H, that can directly recognize CMV, mCMV, I should say, encoded product expressed as a cell surface of mCMV infected cells. This is quite reminiscent of a T cell response or even a B cell response. And it's not only that, it's not only because of the clonal expansion and with the dedicated receptor recognizing the target, but it's because indeed, at the end of this clonal expansion, there is an attrition of this proliferation and hence of the size of the cells which are activated. But there is a small subset of cells remaining which have properties of the activated cells. And epigenetic studies have shown that this is really similar. There are some resemblance, I should say, to the signature of memory cells. So yes, sometime it's difficult to be more precise at this stage because it's still work in progress, but one can find subset of NK cells with effector properties that have been induced by an inducer, and these cells remained present and active quite some time after the initial trigger. So this has been shown in the context of microbial infection. To my knowledge, it has not been shown in the context of cancers. It's still work in progress. It has been shown also that this subset of cells can be induced by cytokine, in particular a cocktail of cytokines, IL-12, IL-15, IL-18. And I'm sure that Katy will come back to this because she's using this cocktail of cytokines to grow cells for NK cell infusion into patients.
Okay, now if I may, the last questions, the basic of NK, you tell us that you can differentiate different subtypes of NK cells by producing different chemokines and cytokines, but what exactly is their mechanism of killing? I mean, we know the canonical mode of killing, but you said that they produce cytokines, which may induce, I don't know, ferroptosis or apoptosis of the cells, or just recruiting other cells, or do we have more insight now the mechanism of killing above and beyond the classical mechanisms of
First, thanks for the question. So first of all, we should maybe remember to everybody what you call the classical way of killing. So this is a granule exocytosis mediated cytotoxicity, which involved the secretion of perforin and granzyme, in particular granzyme A, granzyme B, that are inducing the killing of the cell. Importantly, it has been shown, as you are referring to ferroptosis, so one can talk about pyroptosis as well. It has been shown in a very elegant science paper not very long ago that in fact granzyme A released by NK cells, is also true for cytotoxic T cells, can itself cleave gasdermin in the tumor cells and induce the death of the tumor cells through the cleavage of gasdermin. So the mechanism of killing of NK cells are, as illustrated here, far beyond just what is described in immunology textbook when perforin is inducing holes into the tumor cells, allowing granzymes to have the direct killing activity. Sometimes there are other partners such as gasdermin. That's for one mechanism of killing. The other mechanism of killing include fast ligand and TRAIL, which can be also expressed on subset of NK cells or different type of activated NK cells. And this can also be shared by a subset of ILCs. And this is another way of killing the target. And finally, and thanks for mentioning this, because we even didn't send the paper out so far, so you are visionary. We have identified that gamma interferon by itself, through CRISPR-Cas genome-wide screen, that gamma interferon secreted by NK cells can itself induce the direct killing of the tumor cells. So one should not underestimate the fact that gamma interferon is not only a molecule that is inducing a multicellular immune response of innate and adaptive immunity sides, but also can have a direct effector function, in particular in killing tumor cells.
Okay, thank you, Eric. So now I think we have the good basic knowledge of NK to go into the hardcore of the talk today. So now we know these NK cells. We can use obviously them to kill the tumoral cells, and there is two mechanisms, to activate the cells and generate some cells, and also to engage with antibodies. So Eric, can you summarize rapidly how we can use the NK for cell therapy or to kill cancer cells, and also maybe focusing more on antibody, and because I know you are the specialist of that and the pioneer of this kind of treatment.
Yeah, thanks, Olivier. So I'm sorry I'm kind of monopolizing the talk right now, but I will leave the floor very soon to Katy afterwards. Okay, may I have the forthcoming slide, please? Can you move forward? No, another one. Yes. So this slide is actually here to try to illustrate the main modalities, at least to my knowledge, of NK cell therapies. And as you can appreciate, it comes in two flavors. So the first one, the top panel, is telling us about the monoclonal antibody-based therapies. It's itself divided into two categories, and I'll come back to that. And then there is the cell-based therapies, and all this theme of NK cell-based therapies will be covered by Katy, and Katy will tell us about the different sub-modalities of these NK cell-based therapies. But coming back to the NK cell monoclonal antibody-based therapies, they are themselves coming in two flavors. On one side, this is on your left-hand side, you have an antibody represented in red, which is here to represent immune checkpoint inhibitors, such as monalizumab. Monalizumab is an anti-NKG2A antibody. So NKG2A is itself a cell surface receptor, an inhibitory cell surface receptor that is expressed on a majority of NK cells, plus also a subset of T cells, actually, interestingly, CD8 plus PD1 plus T cells. And it turns out that the ligand for this inhibitory molecule is actually a non-classical MHC class 1 molecule called HLA-E in humans and QA1B in the mouse. And as for PD-L1, PD-1 interaction, the interaction between this non-classical MHC class 1 molecule, again, HLA-E in human NKG2A, is inducing an inhibitory signal within the NK cell. So one obvious possibility to harness the cytolytic and the cytokinic function of NK cells was to generate an antibody that could block the interaction between NKG2A and its cognate ligand. So this is what we have done at Innate Pharma in collaboration with my lab at Center for Immunology and Marcel Lumini. And actually, I'm thrilled to tell you that this molecule, monalizumab, is now in phase 3 clinical trial, not in hematologic malignancies. Sorry, I'm out of the scope of this presentation today, but it's in the non-small cell lung carcinoma. But there are studies going on, clinical trials going on to test the function of this monalizumab. Interestingly, and I think it's a general theme around NK cell therapies, the safety profile is excellent. I think it's a major difference as compared to T cells, and Katy will come back on that because it's also true when you use NK cells themselves as a drug. Now, on your right-hand side, I'm still at the top panel, you have these NK cell engagers. So NK cell engagers are very different from immune checkpoint inhibitors. The idea here is not to unleash NK cells, but it's rather to overstimulate or to stimulate NK cells through the engagement of their activating receptors. So these NK cell engagers have been developed by several groups, and they are coming in different flavors. Just to make it clear, 3 major NK cell activating receptors have served as base for anchoring this NK cell engager. First of all, CD16, which is an FC receptor, an activating FC receptor. A second possibility is based on NKG2D, the activating receptor I talked about. And finally, the other molecule on which we are focusing our NK cell engager approaches is NKp46. But in all cases, the idea is actually to create, through the beauty of the Lego of monoclonal antibodies, to assemble this part of antibodies to create a bridge, a physical bridge, between NK cells on one side and the target cells on the other side. And so on the NK cell side, again, one can try to engage CD16 or NKG2D or NKp46. And on the tumor side, one engages a tumor antigen. So maybe if I can switch to the next slide, there is a short movie. I should apologize to Katy, who has seen this movie 20 times at least, 20 times already. But okay, so if you can come back, because I have no time to, if you can go back to 0, to the start of the movie. Okay, so one start and stop it. It's the ideal would be to start it and to stop it right away. Thank you very much. Wonderful, thanks. Okay, so we start here for these NK cell engagers that we are calling antibody-based NK cell engager therapeutics. We are starting with 2 kinds of antibodies. First of all, on your right-hand side, in green for the variable part of the antibody, antibodies that recognize NKp46. The idea here is, again, to anchor our strategy of enquête on NKp46 for 3 different reasons. The first one is that it's an activating receptor. So the engagement with the antibody will induce NK cell effector function, both in terms of cytotoxicity, but also in terms of cytokine and chemokine secretion. Second, I think is by far the most selectively expressed receptor on NK cells. And third, and importantly, the self-surface expression of NKp46 is conserved at the tumor bed. And it's not the case for NKG2D. It's not the case for CD16 through mechanism that we know about, and we can debate about that in the Q&A session, if you will. But that's one part of this NK cell engager. The second part, on your left-hand side, with blue variable regions, are antibodies that recognize tumor antigens. So we know of many tumor antigens. We know CD20 in non-Hodgkin B-cell lymphoma. We know also CD19. I'm sure that Katy will come back on that. But we know also another molecule, HER2, EGFR, Nectin-4, and so forth, and so forth. So what we are doing here, and now you can put the movie on again. Juliette, if you can, push the button. Here we go. So we are manipulating these different fragments of antibody to assemble them. So we do that at the DNA level, obviously, to create this NK cell engager that you can see here. And here is a tri-specific NK cell engager with one part in green, which is an FAB that interacts with NKp46, leading to NK cell activation. One part in red, which is just human Fc gamma 1, that interacts with CD16, also in reddish at the surface of the NK cell, which is itself in yellow at your left-hand side. And then you can appreciate that it's linked to an FAB in blue that recognizes a given tumor antigen. So this is a prototypical trifunctional NK cell engagers that have been developed. And actually, I'm thrilled to share with you that the first molecule that we developed in the clinic with Innate Pharma and AstraZeneca, and Sanofi, I'm sorry, oops, and Sanofi has obtained fast-track designation in acute myeloid leukemia. So now we are back to hematologic malignancies, finally. And the tumor antigen that we choose here is CD123, the alpha chain of the IL-3 receptor. Next slide. And to finish on that, we recently moved from tri-specific NK cell engagers to tetra-specific NK cell engagers by adding another moiety to this tri-specific scaffold. And we decided to start with IL-2 for obvious reasons, because IL-2 is a very good activator of NK cells. But we use a mutein of IL-2, a non-alpha IL-2, in order to prevent the activation of Tregs and also the binding of IL-2 to endothelial cells, which unfortunately can express the IL-2 receptor, in particular CD25. So these tetra-specific NK cell engagers are being developed as I speak. And to come back and to finish on the hematologic malignancies, the first molecule that got IND, and actually we got the first patient in as of last month. So the first molecule that we developed in the clinic in collaboration with nobody, it's a proprietary molecule from Innate Pharma, is targeting CD20. We know the field of non-Hodgkin B-cell lymphoma is pretty crowded, but we also think that there is still an unmet medical need after CAR-T, after T-cell engagers, after other assets. And this is where we are developing these tetra-specific NK cell engagers. I will stop here.
Okay, thank you, Eric. Before to go to the cell therapy, there is a question on the chat, and actually I had the same question on. You said what you use here is to try to activate NK cells by using different kind of receptors and recognizing the tumoral cells. And you told us at the beginning that NK cells might be also exhausted by different ligands as the classical, for example, PD-1 and PD-L1. So do you think and that these mechanisms may inhibit NK cell function when you have stress cells which are recognized by NK cells? It is the first question. And is it a general mechanisms to shut down the activity of NK that we can reverse by some antibody against different ligands?
No, that's a very good point. And I'm sure that Katy will have something to say on this as well. Because this is the basis of reinfusing fresh NK cells instead of trying to resuscitate or to hyperactivate the cells, the endogenous NK cells from the patient. But this is a very good point again. And there is a lot of question mark for which there are unfortunately no real answers. But to give you an idea of what's going on, we know that indeed NK cells, upon chronic stimulation, which often happens at the tumor bed, can be anergic. The first example is indeed the recognition of cells expressing NKG2D ligands by NKG2D. At one point in time, these NK cells get paralyzed, and there are some mechanisms that have been proposed to explain this. That's point number one. Point number two is the role of the tumor microenvironment. We know that TGF-beta, for example, can really badly affect NK cells, impact on NK cells, and convert these NK cells in some kind of ILC1-like cells with diminished or impaired anti-tumor activity. Another part of the tumor microenvironment comes from the work of Myriam Merad at Mount Sinai, who has shown quite recently that there is a population of macrophages, these TREM2 macrophages, which are very famous for other reasons, which can actually dampen the activation of NK cells. So just to illustrate that there is indeed, unfortunately, this possibility that NK cells at the tumor bed at one point in time can become much less effector cells as fresh, newly activated NK cells. But that said, one should take into consideration that the renewal of NK cells in vivo can be quite short. So even though the NK cells present at the tumor bed can be exhausted, and still we don't have the clear definition of what an exhausted NK cell is, at least in terms of gene profiling, it would be nice to have a gene signature that really characterize exhausted NK cells or dysfunctional NK cells. I prefer this term, dysfunctional NK cells. We don't have this, but it's possible that new NK cells come in and actually still exert some effector function. And this is why we are developing the NK cell engagers, because we know that these NK cell engagers will encounter circulating NK cells and will be able to modulate the patrolling, so to speak, of these NK cells across the body. And we know that this modification of NK cell trafficking through ANKET stimulation leads to a massive invasion of NK cells at the tumor bed, because once these NK cells with modified homing code and trafficking function enter into the tumor bed, they will stop due to the fact that there is a part of the ANKET that will recognize the tumor antigen. But this is still work in progress to exactly know what are the mechanisms and how one can try to provide some assets to overcome this possibility of dysfunction. I will stop here by saying that this is obviously tumor dependent. Okay, so what is true for one tumor type is not true for another tumor type. We all know that across all assets, across all tumor types, but I would like to repeat, this is also true for NK cells. NK cells in AML, for example, are known to be extremely affected by the disease, but even within the field of AML, it depends of the stage of the disease. It depends of the type of AML. It depends also of the location of the cell. So there is a lot of complexity that one has to try to describe and understand at one point in time.
But when you use an antibody which recognizes an antibody, do you need to have cells, tumor cells which are stressed, or you don't need anymore? You can bypass this stress because you engage some antigens which are expressed, regardless the canonical function of NK cells to recognize stressful cells.
Well, that's a good point as well. I didn't present so far any data on the harnessing of this recognition mechanism in NK cells. What I presented actually very briefly is the fact that through NK cell engagers, one can use the effector function of NK cells. But we are actually studying exactly your point. Can we also use not only the effector function of NK cells, but the recognition mode of NK cells, so that not only NK cells can participate to the control of, for example, through ANKET, of tumor cells expressing the tumor antigen that we are targeting, for example, CD20 in non-Hodgkin B-cell lymphoma, but also take advantage of the fact that NK cells express a variety of receptors that can recognize other ligands on the same tumor cells. And that would be very nice. And actually, we have preliminary data at the preclinical level showing that it can be the case, because it could provide a solution to the escape of tumor cells when they lose the tumor antigen that you target using a given set. But this is still work in progress, and we really have, what would be ideal would be to have single cell analysis of tumor cells across all the tumor types. So TCGA is nice, but TCGA is mostly bulk. So it gives you a rough idea of what's going on, but just a rough idea. If we would have single cell analysis of the tumor atlas, that would be wonderful, because in particular, coming back to NK cell therapies, that would allow us to identify the indications, or even within a given indication, the group of patients who would benefit the most of NK cell therapies, because, for example, those tumor cells would express ligands for NK cells. But it's a bit complicated, and I will stop here, because if these tumor cells would express ligands for NK cells, the theory would tell us that they would be eliminated. So maybe what we are dealing is patients who would have the editing of their tumor by the different parts of the immune system, and NK cells included. So maybe at the end of the day, we are facing tumors which have already evaded to NK cell recognition, and that's good that we're trying to harness them using different assets to reinitiate their capacity to eliminate the tumor cells.
But you may by taking to induce some kind of favorable environment to reinduce stress antigen.
Yeah, that's sure. But then the question of toxicity as well that we have to face. But yeah, that's a good point. So what we are trying to do by bringing IL-2 is actually not to work really on the tumor cells at all, but to make sure that at least the fitness of the endogenous NK cell that we want to stimulate is optimal. But again, in some conditions, it will not be the case, and that's why there is really a complementary approach using what Katy is doing, meaning re-injecting, armed, fresh, and fully activated NK cells within the patient.
Okay, so you made the good transition, because you are tired to talk too much. So I will leave the floor to Katy. So when you have no more, like you said, fit NK, you can maybe restore the function, or take new, fresh NK and amplify them ex vivo like we do in CAR T cells. And Katy, so I will ask you first what you have done, obviously, and if you try to explain to us what are probably and the advantages to use NK CAR cells instead of CAR T cells, because for non-immunologists it's not clear in our mind to say why use NK because we have CAR T. So can you explain a little bit and come back to the beginning of the story, obviously, to explain to naive immunologists and oncologists?
Absolutely. So Olivier, let me first start by thanking you for the kind introduction, thanking you and Eric for really giving me the opportunity to participate in this Red Talk with you. And every time I hear Eric, I just feel like I learned something new. I mean, he's such an icon in the field of NK cell biology. So it's just amazing to be here. So thank you. And if I may then answer your question perhaps with my slides, would that be okay? It might be easier to explain really the differences between CAR T cells and CAR NK cells. Maybe I can just share my screen. Just want to make sure that you can see my screen. Yes?
Yes, it's okay.
Yep, fantastic. Well, these are my disclosures. I'm citing founder of a company called Sienna that focuses on TCR NK cells. I'm going to be talking about that. And also our CAR-19 NK program, together with 5 additional CAR assets, was licensed to Takeda. So very good question. What are the differences between CAR T cells and CAR NK cells? And of course, these are all what we call living drugs, in that rather than giving a small molecule or an antibody to a patient, the process involves collecting, in the case of CAR T cells, lymphocytes from the patient, isolating the T cells, ex vivo expanding them, and manufacturing them to using either lentiviral or retroviral transduction electroporation to express the CAR gene of interest. Now, there are currently 6 FDA-approved CAR T products for 8 different indications, all in hematologic malignancies, specifically targeting CD19, so patients with lymphoid malignancies, and targeting BCMA, patients with multiple myeloma. As you can see from this cartoon, an autologous CAR T product means that the product is made from the patient for the patient. So the whole process usually takes a number of weeks, although now the improvements have been made in the manufacturing process to try and shorten this manufacturing period, but still it often takes a number of weeks, and it's a single treatment, so that really a patient can only receive the treatment that was the cells that were generated for themselves. Our group and many others have asked this question of, well, what if we can have an allogeneic healthy donor, so a product from an unrelated donor, but a healthy individual, such that we can manufacture multiple products, so multiple CAR T, CAR NK, CAR gamma delta, CAR macrophages, etc. And then we can then freeze these and have them available as an off-the-shelf. So then this would eliminate the concerns with access, so using an allogeneic donor, therefore, will reduce the cost of therapy and increase access to the products for our patients. So we already talked about the advantages of NK cells in terms of the fact that in the allogeneic setting, they do not cause graft versus host disease. Killing can be mediated through their endogenous receptors. As Eric very eloquently mentioned, NK cells have these various receptors on their surface that are capable of recognizing stress signals. NK cells can be combined with antibodies, as you heard. And then, as I will show you, NK cells can also be engineered to express a chimeric antigen receptor. So mechanism of killing, even for CAR NK cells, could be manifold. And then going back again to what Eric was mentioning, the risk of relapse, at least potentially through the target antigen loss, could be less using CAR NK cells when compared to autologous CAR T cells, where the only mechanism of recognition is through the CAR molecule recognizing its cognate antigen. NK cells are incredibly safe in that the common toxicities of CAR T cell therapy, including cytokine release syndrome and neurotoxicity, are rarely observed with CAR NK cells. Of course, there are certain disadvantages to NK cells for cell therapy. One major disadvantage is their limited lifespan in the absence of cytokine support. Normal NK cells only have a lifespan of 1 to 2 weeks. In addition, it's really not clear what is the best starting population for manufacturing. If you're going to be starting an allogeneic donor to manufacture a product, well, then the question is, what's going to be your allogeneic donor? Are you going to be using peripheral blood donors? Are you going to be using induce pluripotent stem cells, there are NK cell lines that have been derived from patients with NK cell lymphoma that have been tested. There are NK cells being manufactured from hematopoietic stem cells. We are interested in using NK cells from umbilical cord blood. Now, of course, each of these donors have their potential advantages and disadvantages for use. We use, as I mentioned, umbilical cord blood for our platform for our clinical trials. And back in 2020, we published the results of our first in human clinical trial, where our cord blood derived NK cells, where basically we isolated NK cells from frozen cord blood units selected from our cord blood bank at MD Anderson, were engineered with a retroviral vector that encoded for the single chain variable fragment targeting CD19. Signaling was through CD28 co-stimulation and CD3 zeta. This construct also encoded for the human interleukin-15 transgene so that the cells could autonomously secrete IL-15 to help with their in vivo persistence and proliferation. IL-15 is a very important cytokine for NK cell development and persistence, and it also included a suicide switch based on inducible caspase 9 that could be activated using a small molecule dimerizer called remidicid to induce apoptosis of the cells. Again, this was first in human. We didn't know what kind of side effects we were going to see, and also it was a first in human incorporation of a cytokine gene in a CAR vector. So in the dose escalation part of the study, which we published, we observed complete remissions in 7 of 11 patients with no cytokine release syndrome, no neurotoxicity. And despite the fact that these umbilical cord blood units were HLA mismatched with the recipient, we didn't see any graft-versus-host disease. And as Eric said, this was to be expected. What was also very interesting to us was that we could detect our CAR NK cells at very low levels by qPCR for up to a year following infusion. So the degree of microtolerance had been established in that these NK cells persisted for longer than really we'd have anticipated. We thought that these cells would get rejected. Now, just recently, we published the full data on the clinical trial from 37 patients, including the phase 2 portion of the trial. And what we noted was that around a third of patients remained in remission a year after receiving the CAR NK cells without consolidation. Now, I think it's very important because a question that's always asked about NK cells and CAR NK cells is whether they can induce durable responses. And based on this study, we did observe durable responses in these patients with heavily pretreated, median of 4 prior lines of treatment, and all of them patients with lymphoid malignancies. Most of them had either CLL or diffuse large B-cell lymphoma. But the other very interesting observation that we made was that the choice of the donor that you use for your NK cell manufacturing has a significant impact on the response of the patient. So what we found was that if you manufacture CAR NK cells from cords that we defined as optimal cords, and these were cords that were cord blood units that were frozen within 24 hours of collection with a nucleated red blood count of less than 80 million, and 1-year progression-free survival was close to 70%, compared to CAR NK cells that were manufactured from cord blood units frozen longer than 24 hours post-collection and with higher nucleated red blood count. And this was the most significant, the choice of the cord blood for manufacturing was the most significant factor to determine outcome on multivariate analysis, more so than the underlying disease, etc. And that choice was very important because ultimately, with an allogeneic cell product, what you want to do is manufacture tens, if not hundreds or thousands of doses from one donor. So it's very important to know how to pick that donor. Now, these data are not really that surprising. We all have very different immune systems. We have very different polymorphisms in our immune-related genes. All of us that work in the lab, be it with T cells, NK cells, gamma delta T cells, iNK T cells, etc., we know that there are some donors that are good killers and some that are not. And so having this ability to pick at least the umbilical cord blood units for NK cell manufacturing then led us such that in all our current and future studies, we are only picking optimal cords for our manufacturing. And basically in our paper, we went into the details of why this could be, and we demonstrated that NK cells from suboptimal cords have a signature of stress and hypoxia. Hence, the higher nucleated red blood count in that cord. Something bad has happened, obviously, either peri-pregnancy or peri-birth, whereas those from optimal cords have a signature of effector function, the NK cells. Then we took this approach now to target another antigen. So now instead of targeting CD19, we decided to go after another antigen called CD70, which is a pan-cancer antigen. It's expressed in many types of malignancies, practically all hematologic malignancies, including AML, as well as many solid tumors. Because for us, targeting CD19 really was proof of principle. So then we used the exact same platform. So this time using optimal cords, and again we isolated the NK cells, we transduced them with our retroviral vector, this time targeting CD70, and to target CD70 we use the extracellular domain of the CD27 receptor, which is the natural receptor for CD70. Signaling again was through CD28, CD3 zeta. We kept the IL-15 and kept the safety switch. In this case, rather than giving our patients fresh CAR NK cells, which is what we did in our first trial, we froze the CAR NK cells. And from one umbilical cord blood unit, we showed that we can manufacture around 120 to 130 patient doses. So from 2 banked umbilical cord blood, we manufactured 250 patient doses. The cost of one CAR NK dose for our patients is $600. As I'm sure many of you know, the cost of one CAR T infusion is about $500,000. And this was a dose escalation study that's recently started. Patients who received standard fludarabine and cyclophosphamide. Our starting dose level is just 8 million cells flat dose. Just to put it in context, to a mouse, we give 10 million cells, and then we go up 5-fold with the doses. And this is a PET scan of the patient that received the 8 million NK cell dose, and a young man with Hodgkin lymphoma. He was in the 8th relapse of his Hodgkin lymphoma. You can see in this PET scan. PET allows us to look at disease activity. So all the areas that you see are black are areas of tumor activity, and you can see this patient had disease both above and below the diaphragm. 30 days after receiving the CAR NK cells, pretty much all the tumors gone. The brain, heart, kidneys, and bladder show up the tracer, which is normal, and this is the day 60 post-infusion. What's amazing with this patient, despite this heavy tumor load, including this huge mass in his pelvis, we didn't see any cytokine release syndrome, no neurotoxicity, no CRS, no GVHD, and actually the patient did very well. We've now treated 10 patients, 8 of whom have responded. So of course, we're very excited about this program, and it's continuing. We also have then decided that, okay, so since our NK cells are so good at being redirected, why don't we get them to recognize intracellular molecules? So as I'm sure many of you know, CARs and antibodies recognize surface antigens, but the majority of tumor antigens are actually intracellular, and that they're processed and presented on the surface of tumor cells in the context of HLA. So TCRs, T-cell receptors, are capable of recognizing these peptides presented in the context of HLA molecules. Of course, NK cells don't express TCR. So we asked, well, what if we engineer our NK cells to express a T-cell receptor? And this way we can target many more antigens. Advantages, of course, is that NK cells, because they don't express a TCR, there's no risk of mispairing with any endogenous TCRs. Obviously, allogeneic NK cells don't have locus graft versus host disease. In addition, around 40 to 90% of human tumors are MHC class 1 deficient. Now, going back to the NK cells' innate ability to recognize tumor cells or targets, one of the most important mechanisms by which NK cells recognize their targets is through this missing self mechanism. So when targets lose HLA class 1s, for instance, virally infected cells or cancer cells often lose or downregulate MHC class 1 in order to escape CD8 T cell recognition. Now your NK cells, through their own endogenous receptors, their innate receptors, are capable of recognizing these tumor cells that don't express class 1. So then you'll have multiple mechanisms by which these TCR NK cells can recognize tumor cells. Now, there is a problem here because NK cells, they don't express TCR and they don't express the full CD3 complex, which of course you need that in order for a TCR to signal. NK cells only naturally express CD3 zeta. So we engineered our NK cells to express the full CD3 complex, including CD3 zeta, epsilon, gamma, delta, also a co-stimulatory molecule, also interleukin 15, and then also a TCR. And the first TCR that we went after was NY-ESO. Lu and Rafet Bashar in my group did the work, and we basically demonstrated that our NY-ESO TCR NK cells control the tumor just as well as NY-ESO TCR T cells. This approach now is being taken to the clinic, again using the exact same platform in that we take our banked optimal cord, isolate the NK cells, transduce them with the retroviral vectors for the CD3 complex and TCR. We then freeze the cells, bank the cells, and then the cells are thawed and infused at patient bedside following fludarabine and cyclophosphamide. The first patient with multiple myeloma was treated 3 weeks ago. So far, no toxicities. The patient is doing great, and we're waiting for response assessment. We have a second trial that's open in sarcomas. As Eric mentioned, often a problem that both T cells and NK cells face is the complexity of the tumor microenvironment. Obviously, in addition to problems with trafficking, penetration, persistence, and etc. So one big challenge within the tumor microenvironment, at least with some tumors, such as, for instance, glioblastoma, is TGF-beta, which is a very immunosuppressive molecule, and we demonstrated that glioblastoma is very rich in TGF-beta. And so we decided to use this then as a therapeutic opportunity, in that we took our NK cells and using CRISPR gene editing, we deleted the TGF-beta receptor 2 gene in order to make the NK cells resistant to the effect of TGF-beta in the microenvironment. And you can see in these orthotopic mouse models of glioblastoma, we demonstrated that our TGF-beta receptor 2 knockout NK cells do a great job of clearing the tumor. Now, one problem with patients with glioblastoma is that they also receive corticosteroids to manage the vasogenic edema as a result of the tumor. So we also deleted the gene for the glucocorticoid receptor, which is called the NR3C1 gene. So developed a multiplex CRISPR gene editing approach, tested it in our mice, and then demonstrated that these NR3 3C1 knockout, TGF-beta receptor 2 double knockout NK cells are very good at controlling the tumor. And then we've taken this to the clinic now. We have FDA approval. Patients 1 and 2 have been enrolled. Patient 1 has now received 7 infusions of these cells given monthly with no side effects. So obviously we're very excited with taking this approach to the clinic. Another approach that we've taken is, again, as Eric mentioned, is trying to harness these the effector function of NK cells through CD16 receptors. So CD16 is a very important activating receptor. And so we thought, okay, so NK cells are very powerful. We can redirect their specificity, obviously, with CARs, with TCRs. We've demonstrated that. And there are all these engagers, antibodies, etc., that are available for use. So why not bring together the power of cell therapy and antibodies and engagers? So we combined our NK cells with a bispecific engager called AFM13. This was in collaboration with a company called Affimed. And this bispecific engager binds CD16 on the surface of NK cells and CD30 on the surface of tumors. And we demonstrated preclinically, of course, these NK cells that are combined with this bispecific engager are capable of recognizing and killing CD30 positive tumor targets. We took this to the clinic. My colleague, Dr. Nieto, presented these data at ASH. And basically, again, same approach of taking a banked optimal cord, isolating the NK cells. Here, there's no engineering. The NK cells are just expanded. We pre-activate them with IL-12, 15, and 18, such that we can induce a memory-like phenotype in these NK cells. They're then combined with these AFM13 to endow them with these CAR-like properties. And we demonstrated that in 42 patients with Hodgkin lymphoma that had failed a median of 7 prior lines of treatment, and all of them had progressive disease at the time they came to us, our response rate was 93% with a complete remission rate of 67%. And this was really astounding, the type of response. Again, we didn't see any GVHD, no CRS, no neurotoxicity. And just to put this into context, the bispecific engager, this particular bispecific engager had been tested in a phase 1 trial as a single agent before in a similar group of patients with Hodgkin lymphoma. The response rate was only 13% with no CRs. So this combination of cell therapy and engagers, I think, is extremely powerful. Here, the problem was because our NK cells were not engineered. Remember, there was no IL-15 engineering. The persistence of the cells was short-lived. So we had to keep giving repeated cycles, and in many patients, the disease ultimately progressed. So we didn't have that tail that we had with our CAR-19 IL-15 NK approach. So that led us then to come up with this idea of perhaps turn our NK cells into something we call plurireceptor NK cells. And that's a bit busy, I do apologize, but let me take you through it. So we've already shown that we can engineer our NK cells to express CD3 with a TCR. So CD16 obviously is a very important activating receptor for NK cells, and there is a polymorphism in position 158 of the amino acids of CD16. This is a 158 VV polymorphism that then results in the CD16 receptor binding with very high affinity to antibodies. So we thought, what if we take our NK cells and we engineer them to express the full CD3 complex, including the CD3 zeta epsilon gamma delta, a co-stimulatory molecule, interleukin 15, We had to introduce the TCR because in the absence of a T-cell receptor, CD3 is not expressed, is not stably expressed on NK cells or T cells for that matter. So we picked an invariant TCR T-cell receptor so that we don't induce graft versus host disease, but also then it introduces another mechanism of recognition. And then the high affinity CD16. In this case now, our NK cells are capable through their CD3 to be redirected to recognize cancer cells if you combine them with bispecific T-cell engagers. For instance, there are all these FDA-approved T-cell engagers targeting CD19, for example, blinatumomab, targeting BCMA, targeting GPRs, etc., CD20. Through the high-affinity CD16 now, we can combine them with any monoclonal antibody or bispecific NK engagers. And these also include the ADCs because many of the antibody-drug conjugates still have their IgG. Our NK cells express invariant TCR, so they will be able to recognize antigens in the context of CD1D. They continue to express their own innate receptors, their own mechanisms. They still express NKG2D, DNAM, TRAIL, etc. And then they will secrete interleukin-15, which means they will persist. So we made this product, and then we tested it. Of course, we knew NK cells in combination with antibodies work. The challenge was, would our plurireceptor NK cells in combination with a bispecific T-cell engager work? So we performed the animal studies in a myeloma mouse model where the plurireceptor NK cells were combined with a bispecific T-cell engager called elranatamab that recognizes BCMA on the surface of myeloma cells and, of course, recognizes CD3. So here you can see these myeloma cells, these mice are engrafted with MM1S cells. It's an aggressive model in that by about a month they pretty much all have tumor and they die. This is an NSG mouse model, so immunodeficient mouse model. There are no T cells, no NK cells. If you give the engager alone, elranatamab alone, because there are no T cells, there's no control of the tumor. If you give your plurireceptor NK cells alone, without an engager, obviously there is no antigen specificity, and this model is an NK resistant model, so there's no control of the tumor. One injection of the plurireceptor NK cells together with the bispecific engager resulted in cure of these animals. So of course we are super excited with these results, and now we're in the process of taking this to the clinic, where again we're picking bank optimal codes, isolating the NK cells, transducing them to express the CD3, CD16, IL-15, etc., banking the cells as a frozen product and we're going to have a biobank of these plurireceptor NK cells, which will then be co-infused with the antibody of choice. So basically we thaw and infuse the plurireceptor NK cells, and then we also give the bispecific engager separately to our patients. We've already made all the GMP grade material, and FDA submission is underway, so I'm very excited to hopefully get started with this trial in the very near future. So just to let you know the programs that are currently ongoing in the lab, and I didn't get a chance to go through them. We have the CD70 CAR-NK trial in heme malignancies. I showed the example of the patient with Hodgkin lymphoma. We have one in solid tumors. We have a program targeting TROP2 in solid tumors, both pancreatic and ovarian cancer, where the cells are given intraperitoneally, and in metastatic models of breast cancer, as well as in lung cancer, where the cells are given IV. We have the CRISPR-edited NK cells that are given intratumorally with the patients with glioblastoma. All of these are in the phase 1 trial. I mentioned the NY-ESO TCR-NK trial. We have just submitted a a proposal to FDA to target PRAME with our TCR-NK cells. Again, hopefully we'll get started with that soon. We've received approval from FDA for a CD5 targeting CAR-NK trial in T-cell malignancies as well as mantle cell lymphoma and CLL. And we are now we're very hopeful that we get started with the first patient in the next 6 to 8 weeks. And then we have this plurireceptor NK platform, which I'm very excited about because it's a modular platform that allows a combination, as I mentioned, with any antibody, but also it allows for multi-antigen targeting. So I mentioned the trial with elranatamab for multiple myeloma. We're in the process of also preparing a clinical protocol where for lymphoma we are combining the plurireceptor NK cells with tafasitamab that targets CD19 and glofitamab, which is a bispecific T-cell engager targeting CD20. So then this is a dual CD19 CD20 targeting, and also like many others, we're very excited about using NK cells for autoimmunity, and we're in the process of submitting. Actually, we submitted yesterday the IND to use our plurireceptor NK cells with tafasitamab for systemic lupus and systemic sclerosis. So I think the field is really exploding. There is so much one could do. I mean, our increasing understanding of biology of NK cells, all the tools that we have available in the way we can engineer the cells. But ultimately, I think for those difficult to treat disorders such as AML, which is challenging, or solid tumors, combinatorial approaches will be needed, be it combining cell therapy with bispecific engagers, with immunomodulatory drugs, with checkpoint inhibitors like NKG2A inhibition, like Eric mentioned, etc. So happy to take any questions.
So, Katy, thank you very much for this wonderful and marvelous talks. When we hear you, it seems that you resolve all of the problem of cell therapy, because, and also you use some kind of a diplomatic way to use the TCR in NK cells and use bispecific and engager of NK. So you use all tools to cure the patient. So it is what we discussed before, maybe science fiction, or which should become real today. So, but more seriously, I have few questions for the chat, and one from Estelle, which is very interesting, because you said you use for your construct some kind of T-cell co-stimulatory molecules to have some kind of ETAM activation, and do you use some more specific ETAM from NK machinery to improve your NK efficiency, for example?
Yeah, great question, Olivier. We did actually, for pretty much all of our CAR constructs, we made different versions of testing the different transmembrane domains, different co-stimulatory molecules, etc. And it was very interesting, because our hypothesis initially was perhaps using NK-specific adapter molecules would be more beneficial than, for instance, using CD28, which is a very T-cell centric molecule. And what we found is that certainly DAP10 was very effective at driving CAR-NK activation, but also CD28. And actually, we have a paper that's currently under revision, specifically trying to understand what is the mechanism, why CD28 is such a good co-stimulatory molecule, CAR-NK cells. What we found is that CD28 co-stimulation makes CAR-NK cells behave a little bit like CAR-19 T cells with CD28 co-stimulation. Very rapid cytotoxicity, very rapid engagement, rapid, and also serial killing. And what we found was, despite the fact that NK cells don't naturally express CD28, I mean, there are descriptions of CD28 homologs in NK cells, but with a different ligand. And there are some NK cell lines that express CD28, but mature NK cells don't express it, and cord blood NK cells don't express it. But as I'm sure many of you know, in T cells and T cell signaling, CD28 acts as a docking site for LCK to then come and activate CD3 zeta. Well, NK cells have LCK. So then what we found happens is when you introduce the CD28 molecule in the NK cell, LCK comes and binds to it, activates CD3 zeta, and then the whole downstream signaling happens as one would expect. And then if in our CD28 co-stimulatory molecule, and this was work that was done by Sunil Acharya in the lab, where he went and basically mutated all the various docking sites on CD28 for LCK binding, this effect was totally gone. If we do not have CD28, at least a co-stimulatory molecule in our NK cells, we find that they, over time, to multiple tumor re-challenges, they lose their response much faster than NK cells with co-stimulation. I do think that the choice of co-stimulatory molecule is going to be context dependent, very much based on the tumor that you're targeting. For instance, with CD5, we found that with CD5 CAR NK cells, we found that DAP10 was a very good co-stimulatory molecule.
Okay, so I have one question. It seems very surprising that although the cells seem more efficient than natural T cells, you do not observe any ICANS or CRS. Do you think it's because you have such efficiency to kill the target that the NK could not produce too much cytokines, and because you kill by the classical mechanism of killing of the cells, the tumoral cells, and then you have no time to develop CRS and ICANS, because in T cells probably you have not such a strong tool to kill the cells, so the T cells become activated, and then you have this CRS and ICANS.
Yeah, it's a great question. I'd love to hear what Eric thinks as well. We don't have a straight answer. So the answer is really, I don't know. I can tell you that NK cells, yes, you're right, they kill very quickly. Sometimes with our patients that have, let's say, a lymph node that is visible within 3 or 4 days, we see the lymph node like shrink and disappear. So the effect is very rapid. We know that NK cells don't secrete much interleukin-6, so they have a different, obviously, cytokine profile. But as you know, with CAR T cells, much of the mechanism of this CRS and neurotoxicity appears to be coming from activation of the myeloid compartment. Now, NK cells also crosstalk with the myeloid compartment, but it's very different. NK cells are very good at killing myeloid cells. They are very good at killing activated monocytes, for instance, and they're very good also at modulating the phenotype of macrophages and myeloid cells. And we're trying to understand whether it could be this crosstalk of the NK cells and myeloid cells, which is what is driving this very different safety profile. And I don't know, Eric, if you have any thoughts on why NK, and also with engagers, for instance, bispecific NK engagers are much safer than bispecific T cell engagers, for instance.
Yeah. So I don't know if I can jump in the discussion, Olivier, if I'm allowed to jump in.
If you have your question, I have a lot of more. You said, for example, you may kill also the MDSC, and which may explain the better response in NK when compared to CAR T. So let's jump to the question, because it is late, and we may have questions for all night, but let's go, Eric, to conclude on the pitfall and what we can do in the future to improve the NK therapy.
So I didn't understand. Should I jump in or not?
Go jump.
Yeah. Okay. I'm sorry. I was confused. No, I just wanted to complete what Katy was saying about the extremely good profile of NK cell therapies. And I think that besides everything that she mentioned, to which I concur, I think that also the low number of NK cells is a big difference as compared to the number of T cells. In particular, when one compares the activity, the CRS activity, unfortunately, of some T cell engagers as compared to NK cell engagers, I think that, again, NK cell would represent at most 10% of the total number of T cells, and that probably contributes to the fact that there is no such hyper production of cytokines.
You know, I realize in your protocol you use lymphodepletion, actually with high dose of fludarabine and cyclophosphamide, even sometimes even a bit higher than in some protocol with CAR T cells. And do you know if you do need such lympho T cell depletion, because probably the mechanism of expansion are not the same as T cells? And my second question is, you may lose that you may have with NK the production of cytokines and antigen spreading and so on. And if you have a T cell depletion, this mechanism of action might be reduced. So do you really need to have such a lymphodepletion before using NK therapy?
Yeah, no, great questions, Olivier. The lymphodepleting chemo regimen that we use is very similar to what is used with autologous CAR T cells. So we use 30 milligrams per, let me just see, 30 milligrams per meter squared of fludarabine, 300 milligrams, there you go, and then 300 milligrams per meter squared of cyclophosphamide for 2 days. So 3 days of fludarabine and 2 days of cyclophosphamide. Maybe the write-up wasn't very clear. But you are very right, very correct that yeah, and I didn't include it here. You're very correct in the question of, do we need it? And the answer is, I think you do need some lymphodepletion because our cells are fully allogeneic. And unless you give some lymphodepletion, the recipient T cells will reject the incoming cells. In terms of activation of the endogenous immune system, I think that there is certainly contribution of the endogenous immune system to our CAR-NK cell response. And part of the reason I see that is that the results that we see in our patients are often better than the results we see in our NSG mice. And we've also gone and looked at the T cell and myeloid cell phenotype of our patients after we infuse the CAR-NK cells. And we do see activation of the myeloid cells, and we do see activation of the T cells. We haven't looked specifically at the T cell repertoire to see whether we've induced epitope spreading. We are looking at those. And in our case, I think the mechanism could be twofold. One is the NK cells themselves. Obviously, NK cells are the orchestrators of the immune system. It's always said, the bridge between the adaptive and the innate immune response. But also remember that our vector also expresses interleukin-15, and that IL-15 can certainly then activate the other immune cells within the tumor microenvironment.
There is a question that come back is, in some cases, NK cells seems to do not migrate as well as some other immune cells in tissue, maybe in the CNS or in solid tumors. And then do you think that we can improve that, or do you think that because of IL-15 expression, because of eye activation, they migrate anyway to the tumors and better than the natural NK in the natural tumors?
Yeah, no, great question. NK cells have certain preferential homing places that they love going to. They love going to lymph nodes, and what we've found is all of our lymphoid lymphoid malignancy trials, we've seen incredible responses. There are certain places that they don't tend to go to in sufficiently high numbers for them to be effective, and that's why we looked at it in two ways. One is low-hanging fruit. Can we administer the cells loco-regionally? And that's what we've done in our clinical trial of glioblastoma, where the cells are given directly in the tumor by an Ommaya. And also in our clinical trial in ovarian and pancreatic cancer, we did a lot of preclinical work in mouse models and demonstrated that if you give the cells through tail vein injection to mice that have either ovarian cancer or orthotopic pancreatic cancer, the NK cells will not traffic enough to control the tumor. But when we gave the TROP2 CAR NK cells intraperitoneally to the mice, we could clear the tumor. And so in our clinical trial, the patients are receiving the cells intraperitoneally through a port. But then we also want to try and understand how we can engineer the cells, or perhaps with a different cocktail of cytokines, change their homing and chemokine receptor profile to help with their homing. And also the other approach that we're very interested in is to see whether we can combine radiation at the tumor site with adoptive cell therapy, because again, the tumor microenvironment is very much modulated by the effect of radiation. Release a lot of chemokines that could potentially attract the cells.
And do you have any idea and why you show nicely that depending on the cord blood you used, the efficiency might be 100% to 0. And do you have some kind of pre-selection or preclinical screening that you be sure that you will use a nice cord blood to treat the patient? And do you know what is the underlying mechanisms that explain what cord blood is working so nicely when you compare to peripheral NK?
Yeah, great questions. And we were very interested in this. And actually, a lot of the idea of this good cord, bad cord came from the postdocs in the lab, because they would come and say, oh, this was a bad donor, this is a good donor, just from doing in vitro experiments, you know, how different the NK cells were behaving. So we do have a selection criteria that we can do before the cord blood unit is thawed. So and it's very easy. We basically call our cord bank, and we say to them, we want umbilical cord blood units that were frozen within 24 hours of collection and that have a nucleated red blood count of less than 80 million. All cord blood banks have that criteria. I mean, they collect that information. We obviously are very interested in mechanism. Some of that mechanistic data, as I mentioned, was published in Nature Medicine, where we looked at the transcriptomic profile, epigenetic profile of the cells, and we found that there was huge difference, especially on their epigenetic profile. So it looks as if these cells have been, and the most important difference was signatures of hypoxia and stress in the bad cords, whereas NK cells from good cords had very high evidence of activity of Tbet and ILMES and interferon gamma and ERFs and all the things that you know is good for NK cell functionality. So now what we've done is all of the clinical products that we've infused into our patients, we've done single cell RNA-seq and single cell ATAC sequencing on those, and we are analyzing all of that data to try and get deeper into what it is that is making an optimal cord, you know, a good cord was making it a bad cord. So for practical reasons, we're only now using optimal cords for our studies, and the AFIMED trial in a way for us was great, because in that trial, that's when we first came up with the idea, and prospectively, we only picked optimal cords for manufacturing. Only 2 patients received NK cells from suboptimal cords, and that was before we had finalized the criteria, and neither of those 2 patients responded. And then for all of our biobanks now, CD5 CAR NK, CD70 CAR NK, TROP2 CAR NK, TCR NK, double knockout, CRISPR gene edited NK, we're only using optimal cords for manufacturing.
Okay, so I think it's very important if we want to use outside the MD Anderson to be sure that to select the good CAR-T. I have maybe one final question, because before to leave the talk to Eric coming from the chat, it seems that the microenvironment is important, and as people said, when you have tumors which are p53 mutated, and we know they may change the microenvironment, they may change the metabolism of the cells and so on. And usually these tumors are also less responding to CAR-T, for example. Is it the case for NK therapy, for example, and how do you think we can circumvent this resistance?
Yeah, great question and great challenge. So the tumor microenvironment is a huge challenge. Of course, we went after the low-hanging fruit, which was TGF-beta, but there is so much more going on and the complexity of that tumor microenvironment. And as Eric said, one size doesn't fit all. I mean, every tumor type is different. Even subtypes within tumors are different. And we're putting a lot of effort into trying to understand that, like Eric's group doing CRISPR screens, trying to find out, what are the mechanisms that we could overcome this impact of acidosis, impact of hypoxia, maintaining the metabolic fitness. We published a paper showing how this metabolic competition results in NK cells losing their fitness in the tumor microenvironment, CAR NK cells losing their fitness in the tumor microenvironment. With IL-15, you could overcome that somewhat. But I think that basic science will have to inform the translation of approaches to the clinic, and that basic science will have to be done specifically for each disease type. The good news is that with the advent of big data, access to big data, all the engineering tools that we have available, and the potential of using AI, we may be able to get to these answers much quicker than I would have expected, like a decade ago.
Thank you, Katy, for this nice word and promoting science, which is very good, and we know that we should do that. And now it's going very, very, very fast from basic to clinical work. Hopefully, people will hear us to provide some money for the basic science. So, Eric, now can you tell us a little bit what you think about the future and the future direction, and the pitfall and the advantages of NK? Before we have to close in hopefully, and not hopefully, but we have to close in 5 minutes because
sure. No, I fully understand that we have to close at one point in time. We just have another 3 hours, so that's fine. No, I'm kidding. No, I think that Katy got already a lot of points. I think that there are many barriers to break in order to make these NK cell therapies efficient and accessible. So I think that as Katy has pioneered, not only infusion of NK cells, but the combination of NK cell infusion and NK cell engagers is actually extremely promising. Now, one has also to face a lot of problems, which is the path for registration. And this is not, this is another game. But, I hope that the results will help, obviously, in the approval of such combination. Now, that said, and Katy mentioned also the combination of NK cell therapies, the radiotherapy. I can't agree more. There are also some other combinations to test, and that can be done in combination with NK cell infusion or NK cell engagers, which is also at the epigenetic level. There are very promising data in the T cell world, and you're fully aware of that, Olivier, with EZH modulators, which are a path to follow. I should confess that besides the basic research studies which have highlighted the features of NK cells, a lot of what we are doing is inspired from what has been done on T cells, and that's fine. There is no problem for that. But it also helps us to extract the most promising therapeutic assets to try to combine them with NK cell therapies when needed. So I'm sorry to state here the obvious. I mean, Katy said it all already, but I think that those are what should, the direction that should be explored. So namely combination of NK cell infusion with different molecules, including NK cell engagers. And on the other side, when the fitness of NK cells, of the NK cells of the patients allows it. And it will not be the case at each time. But there are indications in which And there are stages of indication, I should say, in which which the fitness of NK cell will be appropriate and sufficient to allow the treatment with NK cell engagers. And so this has to be explored. So obviously there are many cancer conditions in which we would hope to test the molecules that are generated. And now it's also a question of price. Okay, we didn't discuss this so far because it was only based on biomedical research, but as I mentioned, the path for approval and the price of the clinical trials can be cumbersome, okay, to say the least. You know, when you look at the beautiful development of anti-PD-1s, it has been done by companies who had the resources to launch many phase 1/2 at the same time. It's not always the case for the companies involved in NK cell therapies. So one has to use a lot of translational studies. Katy mentioned AI, and I think it's an important tool. It's a series of tools, I should say, that should help us to identify the best indications in which NK cell therapies can be applied. I will stop here and thank you all for the invitation.
Thank you, Nick.
And congratulations for the talk, Katy.
Katy, do you have any word to add to conclude your part?
I'm in total agreement with Eric. I think the future is bright. I think there is a lot of promise, and I think that ultimately, as I mentioned, it has to be a very strong collaboration between basic scientists, translational scientists, and clinical researchers. But also, we are going to have to work with our industry partners. In the absence of that, taking these therapies to a wider group of patients is going to be very challenging.
So something also I think is very frightening is that when we learn, more we know, more we can imagine some combination, pharmacological combination, antibody and cell therapy, plus probably for AML, and we need some stem cell support to restore normal hematopoiesis. So it's a whole package, and the whole combinations, if we follow the regular path of development of all these drugs, it will take years and years. So I think we have to get some accelerate view and some platform to be able to go faster and in different kind of patients and have a better knowledge, as you said, IF of the microenvironment, to use the best drug for the best patient at the best time. But we do see that we have an increased level of tools, and it's quite amazing that from basic science in NK, now we go to clinical work, and I think that it was fascinating that you are some kind of, you take advantage of T cells, NK cells, antibodies, and you put all in one place to kill the cancer. And I said I like to conclude to that. And you are showing very good results in hematological malignancies. And I said, as always, hematologists are the best. And well, but it's my personal view.
Your bias will let's say.
I have a conflict of interest because I'm a hematologist. So thank you very much for this very nice talk. And we have been able to talk for hours. And thank you very much. And we are very lucky to have two of you, the two best scientists in NK cells in the world, so I should say. And
well, Eric, certainly. I don't know. I would, as I said, Eric.
I was about to say the exact same thing. So.
Okay, so
thanks to Kalim. I
would like also to thank the Kalim teams which organized this very nice Redtalks, and Laurie, and it's very nice. And
Bertrand.
Also is Shad underneath everything. So we have a second episode for probably NK next year, because it's moving so fast that we can talk even for more 2 hours. Okay, so thank you.
Okay, thank you very much. Thanks a lot.
And a good night for the other ones. Bye-bye.
Thanks. Bye-bye. Au revoir.